Yes, sir. We are live now, sir.
Okay. Yeah, good evening, everyone. Welcome to this live session for the course Functional Genomics. We have floated this Google form where we requested the participants to fill in the queries or questions if they have any. There are a couple of questions that I'll take it up, but then we are also live streaming this YouTube video with me, Rashmi Parihar, that this course is also joined. And what I would suggest is that people who would like to ask any question related to the um, content of the course that we discussed in the first 2 weeks may write your query in the chat box that's on the right side of the YouTube. Uh, if you're opening using web browser, so you can write that. So if you are using mobile, you have to figure it out. But if it is the desktop or laptop, you can see it on the right side. I hope all of you are finding this course useful. First, maybe we'll take it up a few questions uh, that are posted on the using the Google form. Before I answer any questions, but I would like to tell that I am here to clarify any query on the academic content if you don't understand some points and things like that. But if it is related to assignments or exams and other such logistics, you may want to write on the course portal using the NPTEL, or SWAYAM site in the discussion forum. So that will be handled by a different team. So we are not directly involved in that activity. Yeah. So good to see there are students who have mentioned that they're finding this course useful. That's the very objective of the course, that it's of benefit to the students who are crediting. There's one question, um, whether this course certification will help in research and so on. So let me answer this question in a different way. Certificates are certificates, whether it is this particular course or even your undergraduate degree or master degree. These are certificates that only tell that you have successfully completed the program. So that alone doesn't take you anywhere. It is your understanding of the topic and how good you have trained yourself to use your knowledge in whichever field that you are getting in. It's not necessary that if you have done an undergraduate or master's degree in bio-related area, your profession would be in that area. It's not necessary. But then these are the, if you do get in, then these are the fundamentals and basics that's really essential for you to learn more and do well in the program. And if you're changing the field, all these analytical skill sets and your reasoning and other abilities that you have learned in any program, it's going to help you. So in that sense, yes, I mean, a certificate is going to tell you that how good you have done this course and so on. So that gives you a platform for you to um, tell your prospective recruiter or higher study institutes that you have been doing courses beyond what a formal degree would offer, that establishes your interest. Then, of course, you should grab that opportunity. And then, whenever there's an interview, group discussion, you have to do better than others. So certainly, that's going to be helpful. Yeah. Um, so there's another question in the chat box. It says that I'm interested in knowing how to find genes involved in disease, given a disease, how do you do that? So it's altogether a different topic. Of course, there is the course are lectures available on the YouTube as well. That is human molecular genetics. But yeah, it depends on what approach you are using for finding a defect in the gene. So there are two distinct forms of human disorders caused by genetic changes. One is the disorder that are caused because of a defect in a single gene. So one particular gene is involved in certain critical pathway, and now the gene has a mutation. That cellular pathway now is compromised. As a result, one could have a disease. Now that's the approach to find that is different as compared to, say, multiple genes, each having certain variations contributing to a given disease. So these are classified as monogenic disorders, meaning disorders caused by a single gene, and the other group is called as polygenic disorders, meaning multiple genes, resulting in a given disease. So depending on which disease you look at, the approach that you use to identify the genetic cause for the disease varies. In classic approach, people use families where there are affected members in 2 3 generations in a family, and use that data as well as the DNA sequence to identify the gene. Now what people do is that you have individuals that are affected, so multiple individuals from same family or different families. You do whole exome sequencing for the genes and identify, is there any one gene that is mutated in all these affected individuals. If that is the case, then that could be the gene that is causing the disorder. If it is a polygenic disease, then it's a much more elaborate approach. We need a large number of um, individuals that are having the disease, and also you need to have as many individuals that do not have the disease who are healthy, and but from the same ethnic group, same geography. Therefore, you can take care of the variations other than the genes, and then look at variations in the entire genome, and through some statistical methods, you identify what is called as risk factors, variants of a gene that could sort of confer risk for developing the disease. It could be 1, 2, 3, depending on your approach, sample size, and so on. So that's the broad scope. But for all these things, you need genomic methods to decipher or identify the genes having the variations. That's pretty much the question. Long answer for your question. Yeah. There is a question called, I really doubt we have only 10% of the original genome. I don't know what you mean by original genome, meaning you probably want to say the sequence that are unique to the Homo sapiens. That's what you probably want to say. I don't think that one should be surprised if you subscribe to the theory of evolution. So we have evolved from the primates. If that is the case, if that's theory, that's what we believe led to the evolution of the humans. If that is the case, that we would have much of the genome sequence that what our predecessors had. So that certainly can be pretty large. Yeah. So I'm therefore, it all depends on for how many generations we have been living after the evolution of Homo sapiens from our ancestors. If that's recent, then you will not expect a huge difference. But if that is pretty, distant, then you gain more variation, because every generation there are variations brought into your genome. And if that variations are not contributing to or are not affecting any of your physiology, then directly, then that variation is selected and then it is inherited. So that is how you gain new variation. That's what makes the difference. But if such variations affected gene function, that will not be selected. So in that context, the unique sequence of human is not something that is surprising, which is, I think, within the expected range. That's what I would say. So there's a question called activators repressors possess the same type of DNA binding domain. Yes, possible. There are different transcription factor may have a overlapping binding sequence, may not be same but overlapping sequence. So if one is present bound to that region, the other one cannot bind to it. So there are examples. So we do have, repressors and activators competing for the same segment of the genome for its binding and gene expression activity. Yeah, you do have. How does human DNA is matched and how do we know the DNA from which parents? Uh, well, I mean, there's nothing called, when you extract the DNA, every cell for every chromosome in every cell, you have two threads or two copies, right? For chromosome one, you have one thread from top to the bottom, and the same chromosome, you have another thread that is coming from the top to the bottom. So one of the two threads have come from your father, the other one has come from your mother. That's why you are what is called as diploids, because the meiotic gametes, each carrying one copy of the chromosome, fertilized to form the diploid, wherein you now have 46 chromosomes. Now the question is, how do you find which one has come from father, which one has come from mother? So we basically look at regions of the genome where there are variations, like for example, single nucleotide polymorphism, or what is called as microsatellite markers, where there are repeats and the repeats have different numbers, right? So for a given region of a chromosome, your father would have an allele or a given repeat, say 8 repeats. Your mother may have only 5, right? So in your genome, if I do and check whether you have 5 or 8, and if you have, now easily I can tell which one has come from your father and which one has come from your mother. So it's very easy. So it's not very difficult to decipher which copy of the gene has come from father and mother. It's easily one can do. So you can map the variations with the father and mother wherever there is a parent specific variation, one of them. So you can map it easily. So that's not difficult. Is it feasible to store the data stored in data server in the form of DNA? Well, I mean, theoretically, there are now groups that are trying to use that kind of an approach, but nothing is, as far as I know, available right now. So, yeah. So there's a question on various disciplines of genetics that are available for Indian PhD program. You have to understand the PhD program. PhD program is a PhD program. It's not, you have to understand that, so it all depends on what topic you're doing. So you may be PhD in say biology or zoology, but still you may do genetics, or you may be PhD in biotechnology, but still you would have done something in genetics. So likewise, it all boils down to what research topic that you have taken for your research. So um, that's why some of the institutes don't really say PhD in X and Y. So they only say PhD. The rest is what you have done for your research, your publications, and so on. So it doesn't matter, what in which department you have done a PhD. So as long as your research topic is in a given discipline, therefore one need not worry about it. Yeah, I mean there are a large number of institutes and universities in which there are faculty who work in the areas of human genetics or animal genetics or genomics per se or comparative genomics. So depending on your interest, you have to really look at such institutes and universities and then see whether the research programs that they offer would help you. So that's what I would recommend. There are a large number of groups that work in this area. So you look at that. You don't really look at the department or institute name. It doesn't matter. So there is something called transcription factor and activator. This is a question: what's the difference between these two? Well, I mean, a transcription factor is a factor that is, say, let's say we can define that is necessary for the transcription of a large number of genes, right? So it's like a core factor that's very much essential. Now, there could be also transcription factors that are present in which can go and activate a large number of genes, right? Not everything, but a large number. Now, therefore, they really may not have a specificity that one transcription factor, one target gene. So it certainly has more than one. Now, the question is that how then um, a transcription factor may activate a set of genes and the level of transcription and so on. So that is where the activators come. So they basically are more specific to a given gene or its promoter, and it binds and it opens up the region and it recruits or help in recruiting the transcription factor. Therefore, the transcription can begin. So that's the difference between the activators. They on their own they will not be able to transcribe a gene, but they are essential for transcribing the gene with the help from other transcription factor. That's more generic, kind of uh, understanding one can give, right? So, even after um, doing this course, someone says that they are struggling to get a good understanding on the difference between genetics, genomics, and functional genomics. Um, well, I mean, if you study from the point of view in genetics, for example, so what people do, they basically look at, try to understand how a given gene contribute to a phenotype. That's genetics, right? So you're always looking at the genes from phenotype point of view. So it could be plant, it could be animals, it could be any other character that a given species offer, or it could be human, it could be a disease, and so on. So you are interested in linking the functional outcome of a gene in terms of the organism's whatever function with the genome. So that's, you try to map, sequence the gene, understand the function. That's I would say genetics. Now, when you talk about genomics, that you are looking at the entire genome. So you're studying not one gene, but you're trying to understand the entire genome of a given species as to how many genes are present, how closely related they are to the other organisms, how the genes are organized in the chromosome, and related areas. That's the genomics. When you talk about functional genomics, it is more understanding the function of the genome per se. For example, the gene may be present but may not be activated, right? So what are the genes that are activated in a given cell type or tissue type? So that's where people try to look at the expression. People try to look at whether the DNA is methylated. People try to look at whether the chromatin is more open or more compact, and how, for example, the environment may play a role in regulating the gene epigenetics. So that's what functional genomics is, right? So you have genetics, you have where you are studying a single gene, functions, and so on. That is genetics. And then you have, sometimes genetics can be studied without really looking at the gene per se. Like Now, I am, for example, a human geneticist. I can try to, um, study a genetic disorders, its inheritance pattern, whether it is recessive or dominant, whether it could be sex linked, and so on. But I may not even sequence the genome or sequence the gene itself. But still, I can look at the phenotype and study, understand how the phenotype and the resulting are associated. The gene is, transmitted and so on. So that's genetics. But in genomics, you are looking at genomic sequence. You are analyzing the genomic sequence, right? In functional genomics, we are looking at the function of the genome in terms of its transcription, in terms of how the genome is organized and how the gene regulation is happening, how and outside non-genetic factors can affect the way the genome functions, right? That's the difference. I hope now you have gotten it here. And someone is asking, what is the present status of functional genome? See, you have to understand that now when you talk about genome, we haven't really understood how the genome functions. One way to look at is that what are the genes that your genome has, and what kind of proteins that they make, and in which tissue it expresses, and so on. So that's one aspect. But then that is very small component of your genome. So the majority of your genome or chromosome has sequences that really do not carry any gene, and they are extremely important for the genome function. Otherwise, it would have been lost long time back. And the reason we have these sequences part of your genome is that you have some functional role to play and very essential role to play. Now, there are a large number of genomic sequences that are not having the sequence for coding any protein, but still they make RNA. These are called as non-coding RNA or structural RNA or regulatory RNA, whose function is not very well known. So therefore, again, there are a lot of studies to do. Now we are trying to understand how the sequence help in the genome to organize the way in the cell, because the cell is not like a loose bag where all the chromosomes are packed, or the nucleus, where it is packed. Each chromosome has a distinct territory or regions in which it is anchored. And that anchorage within the nucleus is important for the chromosomes to function. So it's always a dynamic, a kind of a status of the genome within your nucleus for every cell type, depending on the function it does. So, one has to have the means and ways to study the genome in the cellular context. So that is something that we are yet to really get into that kind of domain. So that unless you get in there in live condition, how the chromosome or genome is organized and which region is opened up and how the transcription happen, what happens to the RNA, and how the stability of the RNA is regulated, and so on. These are all something that we haven't really understood quite well. So whatever we know is from few studies from few species. Most of the transcription translation have come from East. So our textbook is based on simpler system. So, but the biology is far more complex than what you learn from the textbook. So we haven't really come to that stage where you can say that, okay, I've sequenced the genome. Now I will be able to tell you how and what causes a particular disease, where the disease is a result of, difference in the gene function, not just one gene, but multiple genes. So that is something still a long distance we have to travel. Yeah. If we change some genes causing disease, so basically the question is that if there is a change in the DNA or gene, if that is causing a particular disease, and if you change the gene sequence, will it get well, whether it will affect the genome, or that's what I try to understand. Probably that's the question. Yeah, the current approach of correcting the genomic sequences, like you must have heard about CRISPR-Cas and other thing. People try to, correct that's one, but then still it's not something that people have certified as an approach for therapy. That is still a long distance. There are now questions whether the changes that you do is so targeted it doesn't really affect any other place. So depending on what kind of approach you use and depending on what is that mutation, there are different ways by which people try to correct the gene. So if it is a loss of function gene, meaning the gene is deleted, there is no protein. As a result, you have a given disease. So one approach people do is to deliver a copy of a normal gene copy and then allow it to express, therefore it makes the protein. So that is called a gene therapy. So that people try using viral mediated gene delivery in the region and expecting that viral mediated gene copy would stay there for a while and keep making protein, therefore you get better. The other ones, of course, if it is loss of function, or if it is something like change in the DNA, it affects the function of the gene. So people trying to correct that sequence, that still is not something that's gone to, in a therapy mode. That is still a long way. The third, of course, is to, sorry, suppress a gene which should not be expressing in a given tissue. And it expresses, therefore you have the disease, like it could be a cancer because of overexpression of a gene, or untimely expression of a gene, or a gene expressed when in a tissue where it shouldn't be expressed. So can that be silenced? So again, there are antisense and RNAi kind of an approach people try and make, So these are some of those approaches, but not all of them are targeted at the genome. Some of them are targeted at the RNA, some of them targeted at giving an extra copy and so on. So, depending on what you wish to correct, they are different. Yeah, so mitochondrial DNA, there's a question. So certainly mitochondria has its own genome. And that's very, very essential because some of the proteins that are present in the mitochondria are coded by the mitochondrial genome. So if you have defect in the mitochondrial genome, mitochondrial DNA, which is not your chromosomal DNA, these are circular DNA like plasmid present in the mitochondria. If they are defect, then there are disorders caused by mitochondrial DNA. So mitochondrial DNA is essential. These are extra chromosomal DNA, and defect there causes disease, right? So yeah, you certainly have extra chromosomal DNA, meaning genome beyond your chromosomal DNA, which is present in the mitochondria. And there are other DNA in your body, that is a microbe. So your body is full of microbes. For example, your skin has got microbes. Your respiratory tract has got microbes. You have gut. Your intestine has got microbes. So there are everywhere microbes, and they have their own DNA. And all these microbes are essential for you to be having a normal life too. So if you have loads of antibiotics, it kills your microbe that you have. You may have problem. That's why people give what is called as a probiotic. People give you the capsule, or there are tonic kind, so like a yogurt kind. It's available. You take it just to spores, to populate again your intestine with certain microbe and so on. So it's they also have something important for your body. So they do have metabolite that help you and so on. So they all develop over several generations that you acquired because of the place that you are in. And some of us received it from our mother at the time of birth and so on. So you can read about it, and there will be There will be lectures on that little later in this course. Yeah. So can you please explain the process of splitting a species into two separate taxa based on difference in the genome? I do not think that um, um, we we classify the species based on the genome. The genomic classification is done long time. Not sorry, the species classification is done long time back. It is all done based on morphometric analysis and many other you know observations that are there. So this classification is it's done long time back. There are hardly any species that we classify now. The only thing that we are classifying now using genome sequences, the microbes present in, for example, different soils or in your skin and things like that, which you really cannot see. These are microbes. So and it cannot be cultured either because there are no defined medium where you can culture. Because if you want to culture any microbe, so you have to isolate the microbes, make them pure forms by taking one colony and growing them and characterizing. Uh, so that's not possible for some of these microbes that are present in certain soil or some skin and so on. So what people do is that directly you isolate the microbes and sequence the what is that regions of a genome that show variation, like rRNA, the gene coding for that, and so on. And then you look at the variations and try to do a phylogenetic analysis and try to place them in certain segments, like so which taxa it belongs to. So you probably will be able to place them in certain positions in the classification. So, but you will not be able to really, go beyond that because now people start sequencing the DNA or part of it, and then you try to classify them. And since each one of us, our skin microbiome, for example, could be unique. You have hundreds of different species, so it's going to be rather more difficult a challenge to identify them in different groups right now. So it still isn't something evolving. It's much more complex. So you'll not be able to sequence everything, but they just started all these things. Yeah, yeah. Once you have a good database for all of them, then you'll be able to do sequence analysis and then classify. Yeah. Yeah, somebody asked about the sequence variation. That's what I explained. So it's all based on the sequence variation only because otherwise you are not able to get the isolates, right? So only you sequenced each of this single organism, a DNA sequence, and then you look at regions that are not conserved, like there are hypervariable regions in the genome. So that segment you sequence because then only you'll be able to look at an evolutionary distance. So normally these are done using that kind of a hypervariable region because the conserved regions, mostly these gene sequences are conserved. Then for evolutionary purposes, especially if you want to really look at the phylogenetic analysis or position of the species, then you look at the hypervariable region. So that's how you do that. Yeah. Now, someone asked for the transcription. I have already explained that, activator versus transcription factor. Maybe after this class, we can go back to the YouTube and then play it again. So I already explained here. Well, I mean, if someone is interested studying more, then you can really look at review articles. There are very good review articles in the field. Functional genomics is again a vast area. So whether you are looking at epigenetics or genome or transcription, transcriptome, phylogeny. There are different segments within this, depending on your interest. So you can really look at review articles, and then many of these review articles are available in journals that are freely available. So these are called as open access. So there is a site called PubMed, or National Center for Biotechnology Information, or you can give a Google search called NCBI. And then if you go to that, there is another link called PubMed. So in that, if you go and give a query and the PubMed, and then it will list all those papers that talk about a given topic. And then in that, you can choose review articles and then see which are the review articles that are available, to anyone without any subscription. That tells you it's available, full length available, full text available. So you can go and read. And if your university or institute has the access, well and good, you can go and read more. So I think that's the best way to do, because textbooks are not something that, because this field is evolving so fast that you don't have a textbook that is comprehensive or up to date. So best thing is to read about the emerging areas, topics using review articles. That's what I would recommend. Yeah. The question is, I am a last year Doctor of Pharmacy student. Can you please suggest some internship? Yeah, I mean, that is something that you need to ask yourself, what is the area, the research area? I mean, you say you're doctor of pharmacy, you're a PhD. If you're a PhD, I don't know whether you really need to have an internship, but if it is B.Pharm course, I mean, you ask yourself which area you would like to have an exposure, and then next step is to look for labs that work in this area in the country, and then pick up some 10, 15 labs and write to them with your CV and a reason as to why you would like to have an internship. And whichever lab has a vacancy, they might, write back to you. So that's the step. Yeah. How DNA sequence can be managed if hybridization is taking place? I really do not understand the context. Uh, in this question. So, are you referring to any techniques, or um, I don't know. Maybe you want to rephrase the question here. Yeah, there's a question on which field of genetics um, one can opt for in the masters. Yeah, there are multiple programs. There are programs that offer biomedical genetics in some universities. Some universities give genomics. Some universities offer masters in biotechnology, but they may have courses in this area. So it's a they are pretty large number of um, programs wherein they do teach genetics. So you can really look for and look for places that have a good number of labs, because masters nowadays you may want to do some thesis work or project work. So if you can continue doing that in the same university or institute, that's quite good. So you can certainly look for. I don't know if there are more questions, then we can look at it. Yeah. I would request that you ask questions that are relevant to the course. This is meant for discussing the topic that has been covered so far in this field. Yeah. Yeah, there's a question whether there are opportunities in industry and so on. So yeah, there are, of course, yeah. Well, I guess we have covered most of the questions. We'll have one more live sessions. Um, um, we can take up more questions in the next live session as well. I cannot really explain the knockouts, um, how to create knockouts in the live sessions. This is something that you have to go and read or listen to the lectures again and read them. So, explaining this here would be almost like giving a lecture, right? So, the knockouts are there are conditional, there are global. I would recommend that you go through the lectures again, and we do provide some links for further reading. You certainly do that, and then we have two PMR of TAs that are there, teaching assistants that they may be able to tell you more on that. So that is there. Now it would cost me another 1 hour lecture, so that is not something that I can explain here. Yeah, knockout. Yeah. I think that's all for today. I guess. Thank you all of you for joining. So maybe we'll meet again 2 weeks from now, and then address. Yeah. Thanks, Murli. Yeah.
Thank you so much, sir, for your valuable time. I'll just end the YouTube stream now. Thank you,
Yeah.
Rashmiji. Bye, sir.
Thank you.
Thank you.